thank you very much for the great invitation to join the MedicReS International Congress on Good Medical Research. It's an honor for me to participate in this. I've been doing clinical research now for many years and would love to share with you some of my thoughts on this. Primarily, I look at research as different ways. I look at either, you're either a clinician who does research or a researcher who does clinical work. It's really hard to be equal with both. For my perspective, I'm mostly a clinician who does research. So things that I will tell you are a little bit tainted or biased in that direction. I'd like to focus a little bit on atrial fibrillation research only because that's my field and I believe it's important. And just to put everybody on the same framework, you may or may not know it's the most common sustained arrhythmia in adults, affecting 2.3 million people in the United States, over 4 million in Europe. The prevalence is rising rapidly, and the majority of strokes, at least in the United States, appear to occur in atrial fibrillation patients. Hospitalizations are greater than any other arrhythmia. And you can see on the bottom screen, blood clot in the left atrium that predisposes to stroke. And you can also see the graph of projected numbers of atrial fibrillation rising over time. So this is definitely a clinical problem. When studying this type of problem, you need to identify that there's some challenges with this. And the challenges with atrial fibrillation are similar to other challenges in cardiology, in that it's a heterogeneous disease. It's not necessarily a homogeneous disease. To give you an example, atrial fibrillation in a 75-year-old with hypertension, even if it's paroxysmal AFib that comes and goes, is different than AFib in a young individual. So let's say you have somebody who's 50 years old, healthy, has AFib 5 hours a day, every day, and you have somebody who's 75 with hypertension, heart failure, who has AFib 5 hours a day, every day. Even though the disease is manifesting the same way, these are really different diseases in many ways. Our classification of atrial fibrillation is artificial, and that becomes a problem when trying to study it, because classification is one way we can study a disease. So we classify AFib by the duration of the episode rather than by any real physiology. For example, episodes that spontaneously terminate are called paroxysmal. But again, as I showed you, paroxysmal episodes can be different. 55-year-old healthy, 68-year-old with obesity and obstructive sleep apnea, very different physiology causing AFib, but the classification is the same. And this has impact in terms of when you're trying to think of a clinical study, in terms of who you include, who you exclude, and so forth. In addition, there's less than ideal choice of medications when you're trying to do studies looking at different therapeutics with AFib, comparing them to medications. There are real challenges with the medications. The medications have different mechanisms of action. They suppress AFib modestly, not very good. And one of the medications, amiodarone, has a particularly very long half-life, which makes elimination of it difficult and studying of it, studying with that particular drug difficult. In addition to that, the primary procedural we have for atrial fibrillation is called ablation. And ablation has challenges too. There's an issue with what we call the durability of ablation. If I do an ablation on the pulmonary vein, is it going to be durable? How successful am I going to be? There are also multiple choices for ablation, different ways you can do it, with freezing, cryoablation, radiofrequency ablation, laser ablation, new things coming, and how can you study these? And then, although people do the basic ablation, which is pulmonary vein isolation, a lot of people look at other things and do ablations. They ablate the posterior wall of the left atrium, fractionated signals, the atrial appendage, coronary sinus, scarring areas. And so how do you make all these heterogeneous things into a study becomes a little bit of a challenge. And also understanding that every single one of these ablations may not be durable. You might think you did a full ablation, but when you come back, you see that you didn't. When I think about what types of questions I want to answer, do I want to answer a clinical question, impact of ablation on mortality, hospitalization, for example? That's what we call outcomes research. One of the areas I do a lot of is new technology research, new catheter or mapping tools, how is it going to affect what we do in the future? I've done a few of these studies where we do first in human research, and I'll show you one example of that. New techniques, new things that we learn, can we make things faster, better, safer, and lead to better outcomes? And these are things that warrant research. And then lastly, mechanistic questions. And these are things I don't do that much, but it's important. So is there a way to evaluate what is the mechanism of a disease like atrial fibrillation? Is there a way to look at what's the focus of it, or what are the triggers of it? And that's harder to do, but still a good area for research. Another consideration for clinical studies is how generalizable will you be? And this cuts across all clinical research. This really depends on the inclusion and exclusion criteria, the number of patients evaluated to randomize a single patient. So for example, if the exclusion criteria are quite broad and it takes 1,000 patients evaluated to randomize one patient, then you really don't have a study that's very generalizable. Another issue is the granularity of the data. You can have a study that looks at different outcomes, but you don't really know all the main details. All you know is the demographics of the patients, but you don't know when they started their medicine, when they stopped their medicine, what their dosage was, all of these little things that are very granular that can help you understand more in depth. And so big studies can be generalizable, but may lack granularity, whereas small studies can have a lot of granularity, but lack generalizability. So you need to consider the size of the study that you're looking at based on how much data you need to collect. And you also need to think about recruitment. How easy is it to recruit patients to achieve this? And there's certain studies that no matter what you do, it's hard to recruit patients for it, and I'll give you an example, and therefore you need to look at surrogate endpoints. This is an example of a study that was recently published. This was called the Cabana Study. This is a very famous study. This was published in JAMA, and this looked at a large number of patients and evaluated them for outcomes with atrial fibrillation ablation. In the initial study design, they looked at outcomes such as mortality. Mortality was considered a separate outcome. But what they realized was that they were not enrolling. You can see here in the study text, they say, due to slow enrollment, they had to modify the trial design. So instead of making the outcome mortality, they made the outcome a combined outcome. And that reduces a little bit the power of the study. But again, that's the only way to get it done. This is my personal experience with this study. I was one of the study centers, and it took us about 1,000 patients screened to enroll one patient. And that tells you, when you see 1,000 patients screened to enroll one patient, that this study may not be as easily generalizable, because you can't just enroll quickly in this kind of study. Granularity of the data, I mentioned earlier. This is just an example of granularity of the data. This is from an ablation study. Where they're looking at a particular catheter. This is called a contact force sensing catheter. And you can get here in the table that I show you the typical demographics that you might see, which is fine. But in addition to this, what they also did was they looked at every single ablation, and they collected 55,000 ablation lesions, how long, how deep, how much power, how much force. And so this is a lot of data. They haven't started to look at it, but you have a lot of granularity with this. You can actually start to talk about things that you wouldn't be able to talk about with other studies, because they were able to collect this level of data, this detail of data. Size needed for recruitment issues. So for example, when you look at some of the complications we see, one of the dreaded complications of AFib ablation is an esophageal fistula. The risk of an esophageal fistula is approximately 1 in 1,000, but based on some data that we see. Now, let's say I have an intervention that will reduce this. How many patients do I need to include? So it would be a very, very large number of patients that I would have to include in a study, that I would have to follow to figure out whether they have an impact on this very, very small outcome. So for that reason, what you tend to see when you don't have, when you have a small risk, something like this, is surrogate outcomes. For example, this is a surrogate outcome, esophageal temperature alerts. If you put a temperature probe in the esophagus, how many times does the temperature go up? That's one way to look at it. That's not the same as a fistula, but it's believed that if the temperature goes up, your risk of fistula goes up. This is a modeling, computer modeling study. So again, you don't have the ability to recruit this number of patients, but you can model it. And another one here, thermal imaging of the esophagus. Again, just a way to get to this question without having to recruit this number of patients. Different types of clinical studies, case series, retrospective analysis, multi-center, unique and novel ideas, and then industry-sponsored device studies. Again, I've been participating in just about every single one of these. Case series is helpful to bring a new idea to light, can be single or multi-center, and it might invoke interest in a randomized or multi-center study. This is the kind of study where if you're able to do it and it's interesting, but still single center, then you might be able to recruit other centers. And there's some challenges with this. This is an example. This is from our center. We found that in patients who had left atrial appendage closure by surgeons, often they would leave a leak. There was a little area where it wasn't closed very well. And so what we found is you can close these by using an Asd Closure device. Now, this is one center or two centers. This is kind of a case series. It doesn't tell you, it's not randomized, it doesn't tell you whether this is better, it doesn't really look at outcomes, it just says that you can do this. And so the next step here would be multi-center, would be randomized, would be looking at different outcomes and different options. Retrospective analysis depends a lot on the database. I recommend for young researchers when they start in their specialty to think about creating a database, think about spending a little bit of time and putting a database together. Learning that skill is actually quite valuable. And you have to sort of think ahead when you do that. What are the different variables that you may want to research and study? Aside from things like demographics, age, gender, things like that, you want to look at what is it that you exactly want to study and collect that data prospectively as you go. And it takes a lot of time to set this up, but once you have it set up, it becomes a lot easier for you to utilize it. Again, here we can talk a little bit about the granularity of the data. The more you collect, the stronger your database is and the more you can do. Now, this is a lot easier at a large and busy center, but if you're at a small center, this may not be as easy. You may have more time to collect more data, but still not easy. This is an example here. This is actually one of our close friends from Ankara who came to Austin and performed this research with us, looking at our database and looking at EKGs and finding an association between fragmentation of the QRS and left atrial scarring. And so you can see here that he took the data that already existed in our database and did this retrospective analysis. Multi-center studies can be unfunded. Those are very difficult to perform because in order to do this, you need a lot of people to be interested in it if it's not funded. More commonly, it's funded by governments, by a society like American Heart Association or American College of Cardiology or any society, or by industry. Industry sometimes sponsors this. It can be as simple as a registry. You want to have a registry of every AFib ablation that you do, and you can get a company to sponsor your ability to collect the data, which is important because then you can have somebody you can pay to do this. Of course, simple is registry, more complicated is a randomized controlled study. This is an example of a couple of multi-center studies that we participated in. So this is one called venous vascular closure system. So after an ablation, we can close the vein using a specific device, and that works very easily. But this was a randomized study from multiple centers that we did, and this was industry sponsored. Another one next to it looks at electrical isolation of the left atrial appendage, and this study was really non-industry sponsored, just done by a group of people who know each other from different centers who work together well. Unique and novel ideas. You need to think a lot about what institution you are at. What is your institutional strength? So for example, some institutions do see a lot of patients with congenital heart disease. Some see a lot of patients with heart failure. Some see a lot of patients with coronary disease. So that institutional strength you can rely on. And you can occasionally partner with industry and initiate research yourself, propose to, say, companies like Medtronic and IDEA. This is one example of a industry-sponsored but also unique novel idea that we did. Stereotactic arrhythmia radioablation, so focused radiation to ablate arrhythmia. So this is something we're able to do because we have the infrastructure to do. It's unique, not every center can do this. So if you have a strength at your center, you can play off of that. This is another novel idea. We had the idea of looking at something, the temperature probes. People use different temperature probes when they do AFib ablation, but nobody's ever compared the temperature characteristics. This is a simple study. This can be done anywhere. You can put it in hot water and graph it. It's not difficult to do. But again, it's the kind of study that is novel, it's unique, it's publishable. So easy to do, simple study, does not require a lot of patience, just requires an interesting idea, that's all. And then institutional strengths I mentioned to you, higher volume, unique cases like transplantation, hypertrophic cardiomyopathy, pulmonary hypertension, valve disease. If your institution has a lot of those, even if you're an electrophysiologist, you might be able to look specifically at AFib in this type of patient population. And you can really look with a lot of granularity in those patients and look at arrhythmia outcomes and techniques. This is in a couple of examples. This is an example from a study I did years ago. I was faculty at Stanford University, and at Stanford we did a lot of heart transplantation. So back in 2009, we published the efficacy of the defibrillator in heart transplant recipients. And you're only able to do this because you have an institution with that particular strength. We also published this study on the histology and mechanisms of cardiac lead and tissue interface when you remove wires or leads from the body, because we had very strong mechanical engineers who were willing to work with us and do some of the analysis. So this is a different way to look at things. Device studies, they can be new devices often tested before market approval or after market approval. Ablation catheters or new imaging technologies are examples of this. Oftentimes you may be asked to do a pilot study. These are typically very small studies, 10 patients, very limited number of operators. And the idea here is to gain insight on how to set up a larger study. For example, I participated in several pilot studies looking at new things. In fact, I'm going in a few weeks to participate in another one to look at a new technology. And so the idea is we take the data from the pilot study, and that's how we build the bigger clinical study. This is an example. This is a RF balloon catheter. So I performed the first procedure in humans, actually not in the United States, but in Paraguay, and then after that in Lithuania. But the pilot study was only 18 patients, and we had 4 operators from different places using it. And after the pilot study, we stopped, redesigned a little bit of the catheter, recommended to the company what to do, and then moved forward. So you can kind of see what we called phase 1 was pilot study, 18 patients. And actually phase 2 was 81 patients. Combine them together for the first study. But again, the pilot study helps inform the company how to modify things, helps inform you how to design the study, the bigger study. This is just an example. And you can sort of see phase 1 success rate is not as good as phase 2, 88% versus 99%. So just to give you an idea that modifications were made and things were improved after learning from a pilot study. Pre-market approval studies, typically, for example, a new catheter, industry sponsored, and this is the industry wanting to get approval in that market. So they want to get FDA approval, or in Europe, they want to get CE mark. These don't always answer a clinical question. These are really designed for industry to get approval. They don't care if you get new insight into the mechanism of something. They don't care about outcomes. What they really care about is efficacy of the device and safety of the device. These are typically multi-center, and the big challenges here include IRB approval and getting the contract from your hospital to allow you to do that. If you're going to participate in this type of study, being a high enroller is helpful because that allows you to be on the publication. So in general, I recommend to young researchers, avoid studies that you will not enroll. So if you have a study and you just don't have the number of patients where you can commit to enrolling, don't do it, because that gives you a bad reputation amongst industry people that you're not a good enroller. This is an example of an industry study. This was the pivotal cryo balloon study. We were part of this study. My name is not here because I didn't enroll a lot. Cryo balloon ablation, first reports North American, the STOP AFib study. Again, the study here is really designed to get approval of the device from the FDA. Now, sometimes the industry comes and says, oh, we have post-market approval. So the device is already approved, but we want to test it in a new study. Sometimes these are useful because these are physician-initiated, very insightful questions that are important and need to be answered. But other times it's just the company trying to get more market share, trying to get you to use their catheter, particularly if you don't use it before, but now they want you to use it. So sometimes these can be a little tricky. So I'll just finish up just telling you a little bit about a recent study that I helped design. So we had heard some background evidence of experimental data that using low ionic irrigation, which is basically half normal saline, in radiofrequency catheters. So the catheter actually has an opening, and the opening allows irrigation. So the saline leaves the catheter and that has an impact on the ablation lesion size. So the idea was if you use normal saline, which is the standard, the lesion size is something. If you use half normal saline, the lesion size is bigger. So we performed a pilot study on several patients and noticed that our ablation times were shorter when we used the half normal saline. But we also noticed that it might be a little more risk when we did that. And so we decided to perform a randomized blinded study. So we had the physician blinded. We covered up the bottle of saline, made sure the physician didn't know which one they were using. And we randomized the patients to normal versus half normal. A lot of issues here. One, we made sure that we had a statistician to tell us how many patients we would need. We had a hypothesis in terms of what the success rate was. Of course, we consented the patients and told them there's a little bit of unknown. We don't know whether this was more risk. And then we have to look at what is the outcome. Are we going to follow these patients for 3 years and see if they have less AFib, or do we look at outcome of safety, speed of ablation, and radiofrequency time? These are surrogate outcomes for success, but those are good outcomes anyways. And this is what we ended up doing. This study is currently about to be published in Journal American College of Cardiology Arrhythmia section. So with this, I'll conclude. Clinical research can be really performed at any center. You don't have to be an academic center. You don't have to be a research center. You can find the strength of your center, utilize that strength. If the strength is creating a database, if you have a lot of medical students who are willing to work, whatever it is, use that strength. If you're a low volume center, if you're a small center, increase the granularity. Look deeply into the patients, and that helps you have an impactful study. Participation in industry-sponsored studies can be very rewarding, but you have to be cautious about some of these. You have to make sure that you're somebody who's willing to enroll patients and make sure the study is something that aligns with what you're interested in. And lastly, you have to understand that clinical studies have many limitations. When you design these studies, you have to understand that nothing is perfect, and that this may not be the final word, and you may have some results and other people may have other results. And understanding this is important when designing the study, understanding the limitation of your study. And with that, I will conclude and say thank you very much for the opportunity, and I hope everybody, all my friends in Turkey, are doing well. Thank you.